Hello everyone. Now we will discuss the molecular basis of cancer. This is the part second of the video. In the first part we were discussing the oncogenes and we discussed the oncogenes at the level of growth factor receptor. Then we discussed about the signal transducing proteins, and now we'll discuss the nuclear transcription genes playing role as an oncogene and the further cell cycle. Now going to the nuclear regulatory proteins or transcription factors. All the signal transduction pathways, they converge to the nucleus where the stimulation of nuclear transcription factors takes place. Now these factors then bind to the genomic DNA and activates the transcription of the genes. If these genes are involved in mutations, this will lead to growth autonomy, which will lead to neoplasia. One of that gene is MYC gene. Now MYC gene is a nuclear transcription factor. Its role is to increase the cell growth upon stimulation by the growth factor. Now MYC gene upregulates the expression of the telomerase enzyme. This enzyme we will study in detail in later videos, but this increases the cellular growth. Also, they MYC also act together with other factors and reprogram somatic cells into pluripotent stem cells. It also increases the protein synthesis by upregulating expression of certain genes, and one of the target gene of MYC is cyclin D, which we'll see in this video. It helps in cell progression. Okay, now this was MYC gene, and the MYC gene to note is a very important gene involved in Burkitt lymphoma, one of the fastest growing tumor. This gene is involved in Burkitt lymphoma. Now going to the last oncogene, that is cell cycle regulators, that are cyclins and cyclin dependent kinase. We know that ultimate of outcome of all growth promoting stimuli is the entry of the quiescent cells into the cell cycle. They want the cell to divide. Now, and in cancers, these genes, the cell cycle regulatory genes, can be it can be dysregulated by mutations or amplification so that cancer cell may grow autonomously. Now we should understand what cell cycle is. Now there is a G1 stage, there is a S stage, there is a G2 stage, and it's a M stage, which is mitotic. Now if the cell is not dividing, it is a quiescent cell. It is said to be in G0 stage. Okay. Then also we should know that cell cycle is regulated by activators and inhibitors. And the activators, the proteins which cause the cell to divide, they are known as cyclins. And there are certain enzymes involved which are known as cyclin dependent kinases. These were the activators. Now what are the inhibitors? The cyclin dependent kinases, they have got their own inhibitors. They silence these enzymes and have negative role over the cell cycle. Now both cyclin dependent kinases or CDK inhibitors, they can be mutated in certain human malignancies. Now for the inhibitors we will go in bit detail, but there are two family of these. There is a CYP family, there is the INK4 family. CYP family contains molecules like P21, 27 and P57, and it inhibits multiple CDKs. You will understand their role later. I will tell you. They inhibit almost all CDKs they inhibit. But INK4 family has a selective role. That is P16, 15, 18 and 19. These have a selective role on CDK4 and CDK6. Okay. Now, first let's see. This is the cell cycle till which we are understanding. This was the G1 phase. This is the S phase, this is the G2 phase, and this is the M phase. Okay, these are the CDKs which we're talking about. These are the cyclins, and these are inhibited by certain CDK inhibitors. Okay, this is clear. Now there is a checkpoint. When the cell is going to divide, there are certain checkpoints. One checkpoint is this one, G1 to S checkpoint, and other checkpoint is G2 to M checkpoint. These are the two main checkpoints of the cell cycle. Okay, now let's discuss about the checkpoints. There is G1 to S transition. It checks for the DNA damage before the replication, and in this various cyclins appear and they play a role. Okay, let's stay till that. Then G2 to M transition. It monitors the completion of DNA replication. When the total DNA replication has taken place, it monitors it and checks that whether the cell can safely enter the mitosis. Now this both transition, they can delay the cell cycle progression, and thus they can provide time. If the DNA is not good, it can provide the time for DNA repair, and then if the repair takes place, the cell can go for division. And if the damage is not repairable, then apoptotic factors are activated and the cell dies. But if the defects, the checkpoints, the which molecules are checking it, they are defective, this will cause the loss of normal cell cycle control, and automatically it will lead to malignant transformation. Okay, now to note that there is we understood about the CDKs. CDKs are constitutively present during the cell cycle. They are present in inactive form. Okay, cyclins, they are synthesized during specific phases of the cell cycle, and they help to activate the CDKs. And then there are CDK inhibitors. Okay, now for to remember it, now this was the G1 to S phase, and here we can see there is a molecule involved. This is retinoblastoma RB gene. This is a tumor suppressor gene. The tumor suppressor gene we will discuss in the next video. These are the tumor suppressor gene. This checks this point. Okay, this point is also checked by CDK4, cyclin D. Cyclin D is also mutated in various carcinomas. It is also checked by CDK inhibitors and mostly of the INK4 family. The P16, 15, 18, 19 in the INK4 family. P16 is also mutated in various carcinomas. Okay. Now this was this phase, this checkpoint. Second was this checkpoint, G2 to M. Here very important molecule P53. Okay, this is a tumor suppressor gene. You must remember P53 is not a CDK inhibitor. Okay, this is a tumor suppressor gene known as guardian of genome also. We will discuss this also along with the tumor suppressor genes. This checkpoint is also controlled by this. Okay, and here the inhibitors are these one. So accordingly, this was the normal cell cycle. Accordingly, alterations can occur at various levels, which can lead to carcinomas. Okay, that's it. Okay, now we can see, we know that this one, that CDKs, these CDKs, they are growth promoting. CDK inhibitors are growth inhibiting. Okay, and if there will be overexpression of the CDKs or cyclins, it will lead to carcinomas, and if CDK inhibitors do not work, it can also lead to carcinomas. Example, so we can see like overexpression of cyclin D genes can occur, amplification of CDK4 genes can occur. These genes will these genes need to be over amplified, whereas genes like P16, which is present in melanoma, needs to be inactivated. There should be deletion. Okay, this we can understand by looking at the cell cycle. Okay, this was about the oncogene. Do watch my previous video also, and tumor suppressor genes will be discussed in the next video. Please do like, share and subscribe. Thank you for watching it.